Which of the following mutations would cause a frame shift mutation? Is it silent, missense, nonsense, or deletion? Think about it. Think about it. Silent just changes the base without changing the amino acid. Missense changes the base so that it now codes for a different amino acid. Nonsense changes base. It codes now for a stop codon. Deletion is taking one base out completely. So the answer is D, because neither silent nor missense nor nonsense will add or delete a base. Since they're not adding or deleting a base, they're still reading in the same 3x3 in the same frame. This is not a frame shift mutation for A, B, or C. Only for deletion, because if you delete, you are going to now change the whole reading frame. All right, we will jump to recombination events. Okay, so this is called genetic recombination or DNA recombination. The word recombination means how to combine. Okay, or in other words, if you bring genes of another organism in, then you have a new combination of genes. That's called recombination. So one organism acquires or gets new genes, and then it expresses the genes so that the trait of the organism now changes. Okay, so in Bio 101, if you have taken it here with us at USC Sumter, you have done an experiment in a biolab where we brought in the genes of jellyfish, of a jellyfish, and put it in E. coli and made the E. coli glow in the dark with the green fluorescent color. So that is recombination of the E. coli. It's now expressing a new gene, the jellyfish gene. Okay. Now, in human beings, it is very hard to get genetic recombination, but in bacteria, especially bacteria, may sometimes happen in viruses also. These three are very, very common: conjugation, transformation, and transduction. So in microbes, they can transfer gene horizontally and vertically. What does that mean? This means that, let's say you start with the cell, okay? There's the cell, and that's the nucleus. In human beings, that cell makes two daughter cells. And so these daughter cells get their genes only from that parent cell. They cannot get it from anything else. Or a parent gives their genes, for us humans, to their offspring. Parent to children. This is called vertical transformation or vertical transfer of genes, okay? Going from top to bottom, one generation to another. Bacteria, so I'm drawing a bacteria here, no nucleus, can also do, parent bacteria makes two baby bacteria. Okay, so it can also go parent to offspring, parent to offspring, going vertical. But they can also do horizontal, as you have seen before. They can do one bacterium to another bacteria in the same generation. You know, maybe it's sibling cell, so cousins, brothers, all that stuff in the same generation. That is called horizontal gene transfer. We cannot do that. So we cannot give genes to our generations in the same level as us. We can only do top to bottom. All right, we're going to watch this video. So I'm going to pause this here and let you watch the video. It's in your PowerPoint link. So get over there and play the video. First, gene transfer. This is known as conjugation. We have talked about this several times already, and you know that this involves a pili. So it's transfer of either a plasmid or a small fragment of the actual chromosome. And the cell that is donating the gene is called the donor cell, and the cell that is receiving it is called the recipient cell. And this is mostly seen in bacteria. The connection that they need is called the pilus singular, pili plural. Okay, so pilus is one, pili are many. So you need that connection. So this particular cell is able to make that connection. Here is the pilus, also called the sex pilus, because this helps in sexual reproduction. And if a cell is F plus, F stands for fertility. And an F plus cell is able to grow a pili or many pili. Okay, F stands for fertility. So an F plus is the genotype of a cell that is capable of making this pilus. So here in the cell, there is a plasmid over here, and it has a gene called F factor that allows it to grow the pilus. It also has a chromosome, but the chromosome is not involved in this example. So this is an example where the plasmid is being sent through. So the plasmid is copied, and it's going to go from one cell to the other very soon. Okay, so these are usually done by gram-negative cells, and they are only done if the gram-negative cell has that fertility plasmid, the plasmid with the F factor in it. Okay, so that plasmid with the F factor gene in it is called F plasmid, fertility plasmid. Only those cells can make the pilus. Okay, so only in gram-negative and only if they have the F factor gene in their plasmid. Who can the recipient be? The recipient can be a related species. It can be, if one is E. coli, the other can be E. coli, but it doesn't have to. It can be another genus. So if a bacterium comes into your body and it's the donor, and there are many, many different bacteria in your body of many different genuses, they can be the recipient. Okay, so mostly a cell without a fertility plasmid will be the recipient. All right, let's see some examples of how this is done. So on the left here, you are going to see plasmid being transferred. So here is a plasmid with the F factor, and the blue is the donor cell, the green is the recipient. So the recipient is F minus because it does not have that gene. And a pilus is formed first, right there, and then the entire plasmid is copied. A second copy is made, DNA replication process, and then the second copy slips through. So look at how it goes through. It opens it up, opens the circular plasmid. It goes through and reforms again. Now this has the F factor in it, right? The gene is in it. So very soon, this is going to change into an F plus cell. Okay? In some cells, the F factor will go inside the chromosome, will be integrated inside the chromosome. So that gene called F factor will enter the chromosome. So this is again a donor cell with the F factor inside the chromosome. When that happens, we call that HFR, which stands for high frequency recombinations. where the donor's plasmid is integrated into the chromosome. So it's no longer on a plasmid, it's inside the chromosome. So now, in this case, look what's happening. The pilus is forming right there, and the chromosome is copied, and the copy here slips through, goes over there into the second one, and reforms again. So this cell is now with the F factor inside its chromosome. It integrates into its chromosome, and it is now F plus cell. Okay, so a portion of the chromosome and a portion of the fertility plasmid are transferred to the recipient in the second case. So both cases are possible. So I'm going to let you pause here, and I'm going to let you watch the video at the bottom. Again, this is embedded in your PowerPoint slide. Now let's look at transformation. In transformation, the gene fragment or the DNA are going to come from outside the cell. They usually come as a fragment, a piece of DNA. And these pieces of DNA are in the environment, in the outside environment of a cell. So they are in the media that they're growing in, or they are in a tube they are in. They could be in your body, and they are in your tissues, in your fluids, in your cells, anywhere. So there are pieces of chromosome floating around somewhere around them. Where do these pieces come from? They usually come in your body from cells that have died. So let's say you're taking antibiotics and you're killing bacterial cells. As those cells die, their DNA is going to be released, and it may circulate in your body. And those chromosomes, pieces or fragments, can be picked up by the recipient cell here. So the recipient cell can pick it up. So they're very, very clever, these little bacteria. They can pick up strange, stray DNA floating around them. And the donor and the recipient don't have to be in the same genus or species. They can be unrelated, so they can pick up any fragment. So when we actually transformed, we did transformation of the bacteria in the BIOL 101 lab. We gave pieces of jellyfish DNA around it, and the bacteria picked up jellyfish DNA pieces. So the donor and the recipient don't have to be related. They just pick up any random DNA. So because they can do this, this transformation has become a very, very important procedure in DNA technology. Anytime we want to send some strange DNA into a cell, we try this method because this is the easiest and the fastest method of getting bacteria transformed with the foreign gene. Now, this cannot be done by eukaryotes. So this is very special for microbes, and especially E. coli does it very willingly. All right, so here is a strange piece of DNA that's coming through. Okay, don't worry about the cap plus and all that. There are transporters that are going to let them in. That's what they are, but don't worry about it. So here is a piece of DNA that's coming through. Now it's inside the cell, and then it's going to be copied and incorporated inside the chromosome. Okay, and now this cell can express that DNA as though it was part of it. So again, I'm going to pause, and I'm going to let you watch this watch this video. This is again embedded in your PowerPoint.